Hello. So today we will be discussing about gene set enrichment analysis. The question that is commonly asked is, what is gene set enrichment analysis? So it's in a very linear term, it's you have a set of genes, and now what you want from this set of genes to look at which pathway relevant genes are overrepresented or enriched. Now, question is, what's the importance of it? It's really, really important. As a biologist and computational biologist, like wet lab biologist and computational biologist, my understanding is our data is only as good as we can actually biologically interpret it. For example, we get a set of data and we want to analyze them. So we have this list of genes, and this list of genes are only as good as we can find out that what actually their functions and which specific functional role they play within our body, irrespective of the other parameters. Now you have a certain disease and you took your samples and did next generation sequencing and found out this sort of genes, and you have a log fold change, p adjusted value, and other parameters. Now you want to see that out of thousands of genes, your understanding is that, okay, I want to see which pathways, the functional pathways, are more enriched from this set of genes. So the very first thing in there is that to run the gene set enrichment analysis. There are multiple tools on the market to specifically address this issue. You can use GSEA, you can use the cluster profiler, you can use FGSEA, and many more tools. So the gene set enrichment, this is the website of gene set enrichment, where you can go and actually, based on the molecular signature database, you can download what's the database of your interest, and based on the GSE version also. Now, question could be that another question is, what actually is the your populations, or what is your samples from? Is it human or is it mouse? First, you need to define them. Second part, what gene sets actually you want to be enriched on their pathways? That's a very, very important question. You can go through each of these and get the details of those enrichment molecular signature databases, specific pathways. And you can see that which one fits you best. And each of these, if you go, what you will see, they have certain list of genes within each pathways. So if you go one hallmark of pathways, like, okay, I am getting 49 of them. So I want to see within this hallmark of pathway, what are the genes? You will see the list of genes within that. You can download them in different format and you will see it in there. Now, when you have this first of the hallmark list of your pathways of interest, and each of the pathway had a set of genes, those genes will be compared with your differentially expressed genes. So what you have done, you already performed your filtering, pre-processing, statistical analysis, either you do the bulk RNA DSEQ or any others, or you do any other things. You got your results where you have the log to fold change, the adjusted value, and those data you have. Now, the suggestion is always to run this specific filtered and p-value adjusted data set to avoid any further conflict if later on that's okay, you are running something that's not actually significant. So just avoid that and first do the analysis and get your differentially expressed statistically significant list of genes with the log fold change. Now, when you have this list, and then you have this specific human gene or mouse gene set and the pathways of interest that you actually want to analyze, then it's your time to select the tools that you want to use for gene set enrichment analysis. As I said earlier, there are a lot of online tools, those you can use. So, or there are couple of them. You can use a cluster profiler, like it's R programming based cluster profiler, also, or first gene set enrichment analysis, or GSEA. There are many in the market, and there is a Python version which came 2022 GSEA. That's also good. There are many things like this, which it up to you. So one of the major interest and popular to the people is first gene set enrichment analysis. So it implements an algorithm for gene set analysis, enrichment analysis, and it's actually available as a Birex beam. It has multiple versions, I think, uh, from 2016 to 2019. And in this beautiful paper, the authors have really explained how it works. So you have a predefined, pre-ranked, uh, statistically calculated gene list with log to fold change and p-adjusted value. Now, second thing, what you want to do is, okay, now I selected the tool and now I want to run the FGSEA, first gene set enrichment analysis. So how does it work? People will give you multiple theory, but I always prefer to go to the original paper. And let's go through this. So what does this say? You have differentially expression analysis, which I have shown you. Then you have downloaded the pathways of collection either from molecular signature database or gene ontology, which is GEO commonly known as, or from Reactor. So after the downloading it, what you did that you thought, okay, I downloaded it now. Now what I want, you run your program in such a way that this differentially expressed genes, the list go through the multiple pathways within these specific pathway collections and the look for the similarity of the genes which could be better associated on specific hallmarks or pathways or gene ontologies. And when you have done that, for example, the code could be available from here or probably from the GitHub also. Let me show you the GitHub also. It's better detailed in here. So you run your program and you, after running it, what do you see? That you get this nominal GSEA value, and then you are performing the multiple testing correction, and which gives you the significantly enriched pathways. So eventually, what you are actually getting, you are getting a figure like this. Now the question is, when you get a specific hallmark of pathway, like program cell death or something like this, and that has come with something like this, what you see in here. It could be hallmark of pathway. It depends which database you are selecting for your reference. So you will get the first thing is, let's explain this figure first. So something like this you will get, right? So in here, you are getting enrichment plot for hallmark inflammatory response. Now this figure has multiple aspect which is very important. It's very, very important to understand, and which is probably you are looking through this tutorial to understand this figure. Let me explain each and every part of it very carefully, which I face personally almost regularly from the people who are curious to understand it. The first thing is the enrichment score. So, what does it mean? This enrichment score specifically says that what is the level of specific this pathway is enriched within the genes those you have found with your differentially expressed in populations. So it's 0, 2.4 and higher. So for example, you get this much. Now the second question is, what is this green line represents? So when you look at this running green line, it indicates one thing, that's within this specific pathway relevant genes of your list of differentially expressed genes, if these inflammatory response genes are present and higher in the overrepresentation, or simply that they are overrepresented within your differentially expressed cell population, then this entire area of inflammatory genes will be upregulated on this area. And if they are low represented or lower represented, then it will be kind of leveled or down. Now, the second question, you have which area of this enrichment score I will consider from here. So to answer this question, I would say, okay, within this figure area, the peak of this figure, the peak of this figure, which is specifically, you can see in here, okay, this is the area where what we will be actually considering the enrichment score out of this. Now, the third part in here is okay, understand that. Now, the third part is so what does this actually the vertical black lines indicate? So, it actually means that the genes is actually supposed to represent the genes within this the relevant to this specific pathway inflammatory response pathways. Now, the more they are present, the more intense they will be on this area, and the lower they present, they will be less in this area. Now you can ask what does actually this rank list matrix means in there. It's actually the rank of the genes represents in there, which actually not always represents the number of genes in here. It's just ranking in there. Now we have what do we explain? The enrichment score, the enrichment area of a score, which will be considered, the rank, and we have discussed about this black lines. Now the question comes when you are writing and you are identifying, okay, what is the enrichment score of this inflammatory response? Will you write these only? No, you don't do that. As you can see within their GitHub also, they explained that this is your enrichment score, and also you need to consider this specific normalized enrichment score. Lots of people write this. The just enrichment score on the paper. It's not right actually. This enrichment score is very important and it has to be normalized irrespective of all the plot you have received from there. And then, as the package itself will be doing it for you, you will get this normalized enrichment score from there. And when you are writing, you are mentioning this normalized enrichment score and p value, which is adjusted. You can see it's here also. It's adjusted p value will be mentioning in there to identify this the significance of it. That is the whole lot of the things to explain your data from the biological context that you got your control and treated. For example, you have a set of healthy donor cells, and then you have the donor which has the inflammation. You got your cells which relevant to the inflammation. You did there uh, for this context, for example, bulk RNA-seq. And then you run those using the DE-seq. You've gone through all the steps, and at the end of the day, when you have done all of it, you want to see within your sets of data, do you have any predominantly enriched or overrepresentation set of genes, those relevant to inflammation? So you go to the FGSEA, you can install it in your RStudio, which the instructions are available in here. You just run the bioconductor and install FGSEA, and there are a lot of details in here. And when you install them, And after that, you go through and run on your differentially expressed gene, which already have the log fold and p-value in there. So after that, you run it and you found this that inflammatory response relevant genes are uh, highly represented. So does their pathway, and as an output, you see this high score of the enrichment of them. And when you get this enrichment score, you normalize them. You got the high enrichment, normalized enrichment score, which is statistically highly significant. And you can say, okay, within my data set, the hallmark of inflammatory response is really high. And that makes your explanation, and which means your biological interpretation, quite clear. That was the purpose of today's tutorial, and I believe it helps to you. This is part one. I'll be presenting the part two, how to run the code in your RStudio or using the FGSEA. And I hope it helps to you. Ask me any question anytime. Take care.